So, beginning with the chapter number 81, which deals with the reproductive and hormonal functions of the male reproductive system. So, let's come towards this diagram, which actually explains all the things over here. So, first of all, we have testes. From the testes, above that, we have the epididymis, through which there is this tube called vas deferens. It is going through the bladder. Behind the bladder, it's going to its ampulla. The portion where the vas deferens meets the ampulla, it is having the area of seminal vesicles, which are bilateral, and after the seminal vesicles, it drains via the ejaculatory duct into this region, or the prostatic urethra. This prostatic urethra has prostate gland on both sides. Then beneath it, we have the bulbourethral glands. Then after that, we have the urethra, which continues outward and it moves outward through the penile portion of the male reproductive system. Then coming towards the diagram over here, this diagram actually shows the cross section. If you cut or slice the testes, so how it's going to look like. So this outer covering is actually tunica albuginea, or the outer covering of the testes. Then we have various septa which are going inside, and then we have the seminiferous tubules which are coiled over here. These tubules are 900 in number according to the text. Then the tubules, they drain into rete testes. The rete testes then go towards epididymis. The epididymis is 6 meters long. It is a coiled tube. It is having its head over here, the body where the sperms are stored for the time being, and then it's tailed, from which the sperms go to the vas deferens. And from the vas deferens, they go to the seminal vesicles, then prostatic urethra, then bulbourethral gland, and through outwards. So this is how the whole male reproductive system is there. And this region is actually the testicular artery, which is going to supply the testes with the blood. So now we come towards this region, which is actually a seminiferous tubule. It is the cross section of the seminiferous tubule. But also we can look at over here, which is actually the lumen of the seminiferous tubule. Surrounding it are certain cells. Then this is the outer epithelial layer, and then these are the Leydig cells, which will be surrounding this area. The Leydig cells, they are actually surrounding the outer covering, or they are found in the region of the blood vessel, where the blood vessels are located. But the seminiferous tubules are inside. They are half meters long and they are 900 in number. And when we go towards the cross section, we find this model. This model has this purple portion is actually the epithelium. Going inwards, these brown cells, these are the first stage ones. They are called the spermatogonium. After the spermatogonium, we have the primary spermatocyte. The spermatogonia develop into primary. Then after primary, we have secondary. After secondary, we have the spermatozoa, and then they are finally converted into spermatids. And between them, we have supporting cells which are present over there. They provide nourishment to the developing sperms. These are called the Sertoli cells, and Sertoli, S is for Sertoli and S is also for support. So they are going to support the whole spermatogenesis process. Along with that, they are going to maintain the blood testis barrier. So coming to this very nice diagram, which actually explains that when there is a fetus, it is having primordial germ cells. Primordial germ cells, they are converted into spermatogonia when they enter the testes. Now in the testes, in the seminiferous tubules, they are stored over there until they reach puberty at the age of 13 years. After that, the spermatogonia, they are converted into a lot of things. Firstly, they undergo mitosis. In the mitosis, the spermatogonia are converted into one, again spermatogonia, which replenish the total number, and second is primary spermatocyte. One, this is going to replenish the number of the spermatogonia, whereas this one is going to be converted into the primary spermatocytes. This whole occasion over here, from the spermatogonia into the primary spermatocyte, takes 25 days after the onset of puberty, and this process is a mitotic process. Coming afterwards, after the primary spermatocyte is formed, it goes into secondary spermatocytes via meiosis 1. First meiotic division is here. After the meiotic division, we have the chromosomes. They are still in their pair form in the chromatid. So we want one chromatid per pair. So that is why we are going to have meiotic division number 2, which is going to convert the secondary spermatocytes into spermatids. So this is the process which is going to convert the secondary spermatocytes into spermatids, and then there finally will be differentiation, and with the spermatids will be converted into mature sperms. But the main thing is the total number of days it takes. For the spermatogonia to be converted into primary spermatocyte, the mitotic process takes 25 days. When it comes to the primary spermatocyte being converted into secondary spermatocyte, the meiosis 1 process, it takes only 9 days, whereas the meiosis 2 process takes 19 days. And then the differentiation process takes another 21 days from the spermatids into the proper nourished sperm. Now we will see and we will look at the structure of a mature sperm. So here we have a very detailed diagram of a mature sperm. It is having a head and a tail. So head is having anterior 2/3 portion and a posterior 1/3 portion. Then we have the tail over here. The tail is having a proximal portion with a neck region that is separating the head from the tail. Then this body and then this further portion. In the anterior 2/3 and in the surrounding region, we have the acrosome. The acrosome is going to have a lot of enzymes like hyaluronidase, like proteolytic enzymes, which are going to help in the penetration of the sperm into ovum. Then we have the vacuole over here. Then this is the posterior to 1/3 head cap. Then we have the neck region. Below the neck region, we have the body. In the body, we have this central exoneme, which is surrounded by the mitochondria over here. These mitochondria are going to supply it with a large amount of energy, which is going to help in its movement. Then we have this exoneme over here, which continues from this to downward region. This exoneme is going to have 11 duplets of microtubules with 2 central singlets. These microtubules are actually, this is the structure of a flagellum exoneme, which actually helps the sperm tail to move. When the sperm is moving inside the male reproductive tract, it is moving with undulating motion, but when it is given its proper alkaline medium, it starts to move with the whiplash like motion, having a speed of 1 to 4 millimeters per minute. And on the outside, this whole sperm is surrounded by a cell membrane covering, which is richly supplied by cholesterol. Now we come back to this diagram again, and we can see Leydig cells over here. I told you they have a very important hormonal function. So what is their function actually? The Leydig cells, they are actually going to be acted upon by the hormone by anterior pituitary gland, which is luteinizing hormone. The luteinizing hormone and Leydig cells, they are pronounced the same way. So luteinizing hormone will act on the Leydig cells and it will cause the production of testosterone. After the testosterone production, it is going to help in spermatogenesis. Look at the location. So Leydig cells are over here, and this portion is associated with spermatogenesis. So luteinizing hormone coming from the anterior pituitary is going to act on the Leydig cells and going to cause the production of testosterone, which is going to help in spermatogenesis. This is whole process. But when testosterone is in excess, it goes to give a negative feedback to Leydig cells and then to luteinizing hormone, and hence its production is stopped. This helps to maintain a normal amount of sperms, that is 120 million sperms in a normal ejaculation. Then we come towards this very different type of cell, which was actually to maintain the blood testis period. This is the Sertoli cell. So Sertoli cell is going to help. Look at its location. It's located where the new mature sperms are being formed. So it is going to help in spermiogenesis. But what stuff is going to activate this Sertoli cell? It is actually the follicle stimulating hormone. So the follicle stimulating hormone from anterior pituitary is going to act on the Sertoli cells, and these Sertoli cells will give the nutrients for spermiogenesis, or from the conversion of the spermatids to mature sperms, that is in the development of the head, neck and tail, etc. And when we come toward the gonadotrophic hormone, or the growth hormone, these two, follicle stimulating and luteinizing, are gonadotrophic releasing hormones. But when we come to growth hormone, it will actually help in mitosis process. And where was the mitosis process? It was in the conversion of spermatogonia into the spermatogonia into the primary spermatocytes. So this portion will be helped by gonadotrophic hormone. This portion coming downwards will be held by the follicle stimulating hormone. And sometimes when we are going to prevent large amount of the sperms to be formed, we do this, that the fertilized cells, they secrete something called inhibin, and this inhibin is going to inhibit the production of the follicle stimulating hormone to regulate the normal number of the sperms. For the luteinizing hormone, to suppress the luteinizing hormone, there was stimulus was the excess production of testosterone, which is going to inhibit it. But for follicle stimulating hormone, it is going to be the excess amount of the inhibin protein, which is going to inhibit the follicle stimulating hormone. And this inhibin protein is actually released by these fertilized cells. So this is all process related to the anatomy, as well as the hormones, as well as the spermatogenesis, spermiogenesis, and the structure of the human spermatozoon, or the human mature sperm. In the next video, we are going to discuss about the function of the seminal vesicles, prostate gland, semen, and all other things, etc., capacitation, acrosome reaction, etc. Till then, keep watching, and if you like my video, please subscribe to my channel. It was a detailed overlook from the book, that is the Reproductive Physiology. And if you want more of such videos and USMLE related and plab related content, please let me know in the comments below.